Hello, my name is Kelly Tatham, and today I want to talk a little bit about stem cell research. I'm going to talk a little bit about what it is and what are the arguments for and against this type of research. So, stem cell research is a fairly new area of research, but already there are laws in place that limit the funding provided by the government for such research and experiments to be conducted. That is why I propose that Congress shall lift all restrictions on the use of human embryonic stem cell lines for research supported by the federal government. First off, what exactly is a stem cell? What is all the hype about? So, a stem cell, by definition, is a cell with the ability to divide indefinitely in culture and has the potential to give rise to mature specialized cell types. The daughter cells produced by a single stem cell can either enter a path leading to the formation of a new differentiated specialized cell. So, as seen here in this diagram, this stem cell can either go down the path of becoming a specialized cell, or it has the ability to self-renew and remain a stem cell, which is pretty unique. Where are stem cells found? Human embryonic stem cells are derived from the isolated inner cell mass, or ICM, of blastocysts. And a blastocyst is what the fertilized egg is referred to at about the 5th or 6th day of development. So the cells in the ICM are destined to differentiate into tissues of the 3 primordial germ layers, which are the ectoderm, mesoderm, and endoderm. And together, these layers will create the adult organism, or the adult human. Therefore, research requires the destruction of an embryo and halting further progression at a certain stage of development. So this diagram just shows a little bit better what I was talking about. Here is an egg being fertilized by the sperm, becoming a zygote. And after a few days of division, it forms the blastocyst, which is where the inner cell mass is found, and that is where the stem cells are found. So in order to do research, the blastocyst must be destroyed and the inner cell mass is you are able to find the stem cells there. So the status quo, as of right now on August 9th, 2001, under the Bush administration, an executive order was put in place that restricted funding for human embryonic stem cell research to just those human embryonic stem cell lines that were already in existence. So under this order, it prohibited the National Institute of Health for funding for the derivation or use of additional embryonic stem cell lines. At the time, the National Institute of Health announced that there were 60 acceptable human embryonic stem cell lines. However, in reality, the majority of these lines were not suitable for research due to a number of reasons, some of which included them not being truly pluripotent, or some had been contaminated, or they could not be shipped for further research. So once again, I propose that Congress shall lift all restrictions on the use of human embryonic stem cell lines for research supported by the federal government. So first I'm going to talk about the arguments in favor of the proposition. And the first one is transplantation medicine. In theory, using human embryonic stem cells should be possible to form a number of differentiated cell types, which in turn could be used to develop cell replacement therapies for a variety of human diseases. And already we have some of these in practice. For example, in leukemia patients, there are hematopoietic stem cell transplants that can be used. Another example is with epithelial stem cell based treatments for burn victims or patients with corneal disorders. Here's just another diagram showing the variety of cells that one single stem cell can become. It can either become cardiac cells of the heart, fat cells, blood cells. It can become a variety of different things, all from one single stem cell. So it's pretty amazing the possibilities that can happen. The second argument in favor of the proposition is about drug development and toxicology screening. So human embryonic stem cell lines are very useful in the study of the toxicological effects that new drugs might have, which is a good thing. Instead of using human subjects, we could use stem cells to see if there's any negative side effects. Human embryonic stem cells are also excellent in vitro models for studies and research on the early events of human development, and can be useful in trying to catch possible genetic disorders that might arise. And my third argument in favor of the proposition is possible cures or treatments for cancer, which is the number 2 leading cause of death in the United States. So using research involving stem cells, whether it be the toxicological effects of certain drugs, possible for cancer, human development, or genetic disorders that may develop in the womb, and stem cell-based transplants, which already exist in some cases. This slide is just going back and showing an example of one of the cases with leukemia patients where the donor donates some of his or her healthy stem cell blood cells to somebody who is having to undergo treatment with chemotherapy and does not have a healthy immune system and can be treated using the donor stem cells. So next I will talk about 3 arguments against the proposition. And the first and biggest argument against stem cell research is probably the source of acquiring the human embryonic stem cells. As talked about, they are required from isolating the inner cell mass of a blastocyst, which is a fertilized egg. And a lot of people believe that human life begins at conception, and so therefore destroying anything after that point is akin to murder, whether it be for religious or personal beliefs. This just shows a full kind of diagram of the stages of development and where exactly after about the 4th day it turns into the blastocyst, which here is where we will find the inner cell mass and get the stem cells from. The second argument against the proposition would be from chimeras. This is a picture of a mythical creature that could be considered a chimera, just with the body of a lion and, dragon wings with a dragon head coming off it. But in reality, in biology, a chimera refers to a person, organ, or tissue that contains cells with different genes than the rest of the person, organ, or tissue. And this could either be due to a mutation, but in this case, it is a result of a transplant of cells, organs, or tissues from another person or from a different species. So the ethical question becomes whether or not science can do it, but whether or not scientists should do it. And then the third argument against the proposition would be payment to donors. How much compensation should a female get for her egg in the research of stem cells? And a lot of ethical issues arise when payment is in excess of reasonable out-of-pocket expenses. For example, there was a scandal in South Korea where the researchers were involved in inappropriate payment amounts, as well as serious problems with the informed consent procedures and high incidences of medical complications. And so informed consent problems, lack of understanding, a lot of women who might be poor, uneducated, the motivation and reasons for the money, and they might not understand how risky the procedures can be. These can all be very negative side effects. So in summary, there are reasons for and against stem cell research. As with any controversial topic. There are sides for and against. Moving forward, I believe that research oversight will be key. I think that oversight committees should be formed that consisting not only of the researchers, but also scientists, physicians, ethicists, legal experts, as well as community members. And consent forms also need to be clear and concise and lay out exactly the risks of the procedures and in layman terms, so it can be well understood to everybody. Alrighty, well, that is my talk on stem cell research, and hopefully one day soon there will be enough federal funding that the opportunities are endless on what could maybe be accomplished using stem cells. And so thank you and have a good day.